There's a big difference. You can see 3 of our team members in Antarctica right now, shivering a little bit in their big red jackets. One big difference between them and me is that I'm in my basement, and this is a Zoom background from my time in the Dry Valleys. So unfortunately, I'm not going to get to deploy this year, but we do have some colleagues here. Let me get my my screen going here, and I want to spend a little bit of, I'll spend a moment having them introduce themselves here. Actually, now I'm realizing that perhaps I need to ask them to do that before I get started. So let me go ahead and stop the share, sorry. Kathy and Anna and Mia, do you guys want to just introduce yourselves real quick to the group?
Sure. So my name is Mia Vanderwilt. I'm a master's student at CU Boulder with INSTAR and the Environmental Studies Department. This is my first time to the Dry Valleys, but second time down to Antarctica. Yeah, I'm excited to talk about it more later.
I'm Anna Wright. I'm a PhD student at CU with INSTAR and the Environmental Studies Department as well. I've also been down to Antarctica once to a different part of the continent, so this is my first season with the Dry Valleys, and me are working on the streams together, so yeah.
And I'm Kathy Welch. I started with the McMurdo LTER in its first year in 1993, and I've been to Antarctica every year since then, except for last year. Unfortunately, we didn't have a field season, but yeah, so I've been working a lot on water and the chemistry of glacier streams and lakes. I can tell my face is already getting cold. So yeah.
Terrific.
That's awesome.
Thanks you all so much. So what we're going to do, we've put together a presentation that we want to provide in sort of chunks. So we have 5 or 6 slides or kind of sections, and I'll outline those here when I share my slides. Give me just a moment to do that. Let me share that. And what we'd encourage you to do, we're going to stop after each one of these segments and try to take 5 minutes to answer questions. So what we'd appreciate is if you all could type your questions into the Q&A. We'll go back through those at each stopping point and take a look at what you've asked, and we will ask that of our colleagues in Antarctica, or I myself will try to answer it one way or the other. The other thing I should just let you know as we get started is that this link to Antarctica, while it's awesome, can also be kind of tenuous. So it's quite possible that we will lose our team members there, their link, and I'll have to sort of endeavor to give part of the talk. So please bear with us if we run into some of those issues, but I'm also going to be the one advancing the slide. So here's our team again. That's me on the left. I've deployed, I guess 14 times or so to the ice. I did my PhD work there in the late 90s, and all of us are with the University of Colorado Boulder, and we're all affiliated with the Institute of Arctic and Alpine Research. So that's what INSTAR is as an acronym for our home institute. And as Mia and Anna both indicated, they're working on their graduate degrees through the Environmental Studies program. I'm a professor in civil and environmental engineering beyond also being part of INSTAR. All right, so here's our layout. We're going to go through these 6 topics, kind of 5 to 7 minutes of each of these little pieces of presentation, and then we'll pause and try to keep about 5 minutes or so for answering questions. At the very end, you can see our teammates down there are sort of shivering. I'll do my best to back cleanup and just answer all the sort of leftover questions, but we might let them go if they haven't already been disconnected, just so they can go warm up, because there's kind of a weird situation where the best connection in Antarctica is outside, halfway up Observation Hill, which is just outside of McMurdo Station. So that's why they are outside. It's partly for effect and partly out of necessity. All right. Okay, so I'm going to just introduce us to the Dry Valleys and the LTER overall, and then I will pivot to a talk on lakes that will come up and be presented by one of our teammates on the ice. So as mentioned earlier by Suzanne, this is the largest ice-free region of Antarctica. It's just under 2% of the land area of Antarctica, and it is a landscape that looks like this. So you see mountains On the right-hand side of this, you see a glacier coming down. And then in the foreground, this is an ice-covered lake. And these lakes are closed basin. We'll talk a little bit more about that in a moment. But they during the height of the austral summer, we have 24 hours of daylight, and we have a positive energy balance on these ice masses that allow meltwater to form. So one piece of evidence of that you can see on the edge of this lake are the moats. We call these moats, these shallow regions right next to the shoreline that melt out with very fresh water. This particular lake might be 15, 20 meters deep, and it has an ice cover of about 3 to 5 meters thick on top of that, sort of shielding it and insulating it from the atmosphere. My arrow went away. So we are an LTER site, and there are 28 of these funded around the world. Because they're mostly funded by the US National Science Foundation, most of them are in different biomes around the United States. You can see there are 4 now in Alaska, spanning from south to north. Some of them are coastal, some of them are more marine focused. Ours is one of 2 sites that are funded to study Antarctic ecosystems. Ours is the inland site, and you can see on the lower right, the little inset of Antarctica that's sort of splayed out. MCM is the McMurdo Dry Valleys Long-Term Ecological Research Site, and PAL is Palmer. That is the other site that is more focused on marine processes and coastal processes. What's unique about these LTER grants in the program through the National Science Foundation is that we get grants in 6-year cycles. And what's unique about that is most research grants that are funded through the NSF are on the order of about 3 years. And we typically might, work with a couple of colleagues to make it collaborative research. But these LTER sites are meant to be bigger. They're meant to be longer, and they're meant to dive further into the processes that make these ecosystems work. So in our case, we're studying this polar desert, and one of the things that we are able to do is conduct some monitoring. So we keep our finger on the pulse of our ecosystem, just like all of these other sites do. And then we also have long-term experiments and short-term experiments that we conduct where we test hypotheses that we put in our proposals. This network allows us, there should be, instead of a question mark, that should be an arrow leading to synthesis. That is to say that we can compare our results on specific experiments or specific monitoring activities to someone at the H.J. Andrews Experimental Forest in Oregon, for example, and look at commonalities and differences to try to drive new discoveries in ecological theory. Every one of the 28 sites should be studying 5 core areas, which is to say that somehow we document and quantify and study response of our ecosystem to disturbances, organic carbon cycling, nutrient cycling, the dynamics of populations and communities of organisms, and primary productivity, which is to say the growth, the use of sunlight and carbon dioxide to build biomass, largely with plants. And in our case, you didn't see a whole lot of plants across that landscape. So we'll talk about the aquatic side of that as we get into this talk a little bit. Our group is big and diverse. You're seeing a very small group of us from the University of Colorado Boulder, but we have 9 PIs from different institutions, and we have a number of awesome students, postdocs, and technicians that are supported to conduct their science to achieve their degree, but also to move our science forward writ large. And then it's up to us as principal investigators, I use the term PI, a principal investigator, the lead scientist, to try to put those pieces of a broader puzzle together to understand how the system works. And this is a great framework within which to develop as a scientist. And we hope that that our graduates, from this program move on to interesting other things across the other LTERs or other earth science and ecosystem science studies. We were established in 1993, as Kathy mentioned, and so we are almost 30 years old, which is really gratifying. I have not been the lead PI that long. I've been the lead principal investigator for about 6 years, but I did my research, my student research as a PhD student in these dry valleys working on the stream team. So this is a polar desert. You can see with the red dot there on the map on the left left, we are located at the edge of the Ross Sea. We are about an hour's flight from an island you can't see under that dot, which is Ross Island, and that's where McMurdo Station is. So our friends are coming to us from McMurdo Station at the moment, and soon they will deploy, they will move over to the Dry Valleys. We'll talk a little bit about that as we go. This is an area that we have a mean annual temperature of about minus 18 degrees C. It gets down to minus 40 in the cold, dark winter. And then during the summer, we sometimes have air temperatures above freezing. We also don't get that much snow. And the amount of snow we get is a water equivalent of maybe a couple of centimeters on the valley floors. However, we do have all these glaciers, and the accumulation zones for those are up in the mountains. So we have very steep gradients of snow that collect. We obviously have to have quite a bit of snow to fuel or supply these glaciers that come down from the edges of the mountains to the valley floor. This is a bit of a confusing map, and there are only a couple of things I want to impress you with. On the left-hand side, we are looking with north pointed up, and we have 4 main valleys of the Dry Valleys that we work in. The northernmost one is Wright Valley. Then we have Taylor Valley, which is where most of our work is based. And then Garwood and Mears are to the south. I also just want to, each one of these dots is indicating a different type of monitoring station or experimental location. And I'll just point out the couple of lake basins here because we'll refer to them later. Lake Bonny here actually has 2 lobes, a west lobe and an east lobe. Lake Hoare is named after Ray Hoare, who was a physicist who worked in the valleys back in the 50s and 60s. And then Lake Frixell is another location. And this is where we have a lot of streams coming in from glaciers up in the hills here and these 2 large glaciers on the edge. So this is a primary site of a lot of our work, but we also have a fair bit of work going on when we can catch a ride down to Garwood Valley and to Mears Valley. So we have meteorological stations, stream gauges, and so forth, sort of spread out across these 4 different valleys. Our conceptual model here is that the 2 big vectors that sort of connect our landscape are water and wind. And so on the left-hand side, you can see the landscape. On the right-hand side, if we put a cartoon together in sort of a cool or low surface energy balance sort of a season, we often get kind of typical meltwater inflow to the lakes, that build up the water column, and we have an ice cover and so forth. But then in a high, and we note these as katabatic winds, they're actually phone winds, but these winds, strong winds that blow down the valleys and move a lot of sediment around. They move a lot of algal mat material. So it's an important vector for moving biology and nutrients around the landscape. And then finally on the lower right, we have a high energy input season where we might have much more stream flow. We see more water scattered across the landscape, this dry landscape, maybe potentially a thinning ice cover on the lakes. And sometimes we have much greater wind events that move material down valley as well. And with, let's see, I'll sort of conclude this part of the talk by talking a little bit about the life in this valley. So in the Dry Valleys overall, it tends to be small. Every now and then we get the random penguin that walks through, but the largest animals out there are us as humans, studying from sort of November to about February out in these systems. So in the lakes, we have mats on the bottom. That's what the lower picture is. And then within the ice column, we also have these algal mat materials that get caught in the lake ice and work their way up through the system. In the streams, we have these shag carpets of microbial mats that also include diatoms in them that we've studied. We'll talk a little bit about that. And then in the glacier ice, we have some of this deposited algal material and bacteria, of course, that photosynthesize and respire in these little pockets that they melt out on the glaciers. We're not going to talk a lot about them tonight. And then in the soils, we have these awesome nematodes, tardigrades, and rotifers that occur. And they're not a huge diversity, but that gives us the opportunity to really hone in on the controls of their life cycle and how they exist and how they interact with their environment. So I'm going to pause there before we switch over to the lakes section. And this is a colleague of ours, Dr. Matt Cohen, who came down with us a few years ago. And I'll pause to take just a couple minutes of questions if anybody has them. Let's see, in the Q&A, So who is Taylor Valley named for, which is interesting coming from GF Taylor. So it is named for Griffith Taylor, who was part of Scott's party in 1911. Is there any work being done by scientists from other countries? We have a strong contingent of New Zealand researchers that work in the dry valleys. And once in a while, in our part of the dry valleys, we sometimes see Italian or Korean groups. Their bases, however, are much further north of our section of the dry valley. So they tend to study their local region of the northern parts of the dry valleys. And then, is there any remote logging to the dry valleys during last season? So a great question. We didn't cover this last season because of the pandemic. We did not get to deploy. There were very few deployments to Antarctica overall. And in part, that was for a variety of reasons, trying to keep COVID off the continent, but also our gateway cities were very, very careful about who they let in and out. And this year, so last year, we did not have anyone deploy. However, we did have some technicians who went out and collected some samples for us and got a few stations up and running that were down for whatever reason. So we did actually get to collect some data, which was fantastic, and some samples. This year, we have this new opportunity with 10 people deployed this year, and this is 3 of them that we have with us online here. So I'm going to stop yammering at this point. It looks like we're out of questions in the Q&A, and I'll pass this over to Lakes.
Mike, there's one important question in the chat, actually. I'll say it out loud.
I'm not seeing that.
Can you explain again how the dry valleys can have lakes and streams? How can they be called dry if they have lakes and streams?
Well, that's a good question. Originally, Scott's group called them the Valley of the Dead because they didn't see any life there. They're dry because compared to the rest of the continent, we don't have stacks of ice, covering the landscape. And generally, you see that the landscape is fairly dry, but we do have these focused locations of snow melt and glacier melt that occur. And so I guess from the Antarctic perspective, we're fairly wet. From sort of a temperate perspective, even me in Colorado, this looks fairly dry even to someone who lives in the semi-arid part of the Western US. So thanks, Alex. Great question. I will pass it off now to our colleagues who are going to talk about the lakes. Let's see. Looking good, Kathy.
Okay, yeah, so I have done some work on the lakes over these years, and Mike has mentioned a few of these things already, but I just want to point out that our lakes in the dry valleys are different in that they are closed basin lakes, which means that water can flow in from the surrounding glaciers, but there's no outflowing streams for most of them. So that means they effectively work like a basin that collects water that flows in. During the winter time, the ice covers get thicker. So normally the ice covers are somewhere in the order of about 3 to 5 meters. And during the summertime where it's above freezing, they will kind of stay about that same thickness. They lose mass off the top from a sublimation and evaporation. And then during the winter time, they will get thicker. So the volume of the lakes are somewhat dependent on the relative amount of what's coming in and what's leaving. And the parts that are coming in, as I said, are from stream flow and then leaving through evaporation and sublimation. So over time, that water balance, you can see this is a plot showing the lake level rise relative to the datum that we set at 1972, just for reference. And some of the lakes are much bigger, bigger than others, or have a more shallow basin versus a steep basin. So here we can see that 3 of our lakes, Prixel and Hoare, have basically only risen about 3 meters over the last 50 years, whereas Vida and Bonnie have risen quite a bit more. And that's really just because the lake basins are a lot more narrow, and so a relative amount of water added will increase the lake levels more in the more steep basins than in the shallow ones. And here I want to point out a couple of things about this plot. So we measure the lake levels at the beginning of the summer and then at the end of the summer. And so this kind of sawtooth pattern that you see, it's a kind of short-term change that happens between the beginning of the summer where the lake levels are relatively low, and then they increase based on how much flow we had. And then the longer term trends really look at like how the energy balance has changed over the long term, whether that's more sunny days or warmer air temperatures through the summertime. During the winter, it's just cold, so we don't really melt glaciers and have stream flow, but in the summer we can get glacial melt and streams flowing into the lake. So that's what we're seeing here with this long-term trend in the lake levels. Mike, do you want to advance to the next? So we've been out in the dry valleys measuring lake levels since the 90s, but people had been out there before us. And one of the earliest data points that we have is from Scott's party that came through the Taylor Valley in 1903. And based on their description of the distance across the Narrows, which is that point you can see on the figure here, this is Lake Bonny, the West Lobe and East Lobe. And Scott's party, fortunately for us, when they walked through there, they measured the distance across the Narrows there. So we can use that as a reference point to see what the lake level was like in 1903. And then additionally, in 1911, Griffith Taylor's party, I believe it was, that went through, they got some photographs of that area. So we can use that as another reference. So we know based on that historic data and then our more recent measurements where we've gone out and actually measured the lake level, that the lakes have come up quite a bit over the last more than a century or so. Thanks. So I wanted to mention, as you can kind of tell based on like, my cheeks are already freezing here. It can be a really harsh environment out in the dry valleys. It can also be a really beautiful, wonderful place to work, but we tend to have shelter to keep both ourselves and our instruments warm when we're out there working. So the contractors will go out ahead of us and set up these polar havens on the lakes, which works out really well for us to have some shelter so we can get out of the wind, and we can even have heat in there. So here we see some of the instruments that we use that we deploy year round to collect samples for us. And those, it's really important for us to keep them from freezing before they get deployed. So we have these shelters and it makes it a lot more comfortable for us to work out there, but also to keep our instruments from freezing and so they can function properly before they get deployed into the lakes. I wanted to point out that we are really studying an ecosystem out there. And unlike other, like other places on Earth where we have a food web, we see the same thing in the lakes of the dry valleys. You know, in the lakes, I mentioned there is an ice cover, but the lakes in the dry valleys are one of the few places where we do have liquid water that exists all year long, even in these really sub-zero temperatures. But if you look here, we see that we have the sun, and that's really our main source of energy to support this, the organisms that live in the lakes. And we have a diversity of different things that live there, bacteria and phytoplankton. And the largest things that we have are things like rotifers. So unlike other food webs that you think about in temperate locations, our ecosystem tends to be all small organisms. We don't have anything as big as zooplankton and certainly no crustaceans or fish. A little bit more about the lakes. They range in size. You can see from this plot, we have the lake depth and then the relative volume for each of those layers. So the biggest lake that we work on in the Taylor Valley is the east lobe of Lake Bonny. And as I mentioned earlier, it's kind of like a more narrow and steep valley. So it tends to fluctuate more as more water is added, whereas Lake Frixell is a much more shallow, broad lake. And then west lobe of Lake Bonny is the smallest lake, even though it's pretty deep. It's a very small basin. And then Lake Horror is somewhat in the middle. And then looking at the plot on the right-hand side, I just wanted to point out a couple things. So that's a temperature profile with depth. And at the surface, you can see that the temperature is really right around freezing. The water that's right under the ice is just right at the freezing point. So it's pretty cold, and then a little bit of light can penetrate through that ice cover, and we see the warmest temperatures tend to be in the photic zone of the lake, so just below that ice. That's where we tend to also see the highest amount of productivity for things that are photosynthesizing. And then interestingly, these lakes, we didn't really talk about this, but this is kind of my area, is looking at the chemistry. So these lakes at the bottoms, they tend to be hypersaline. They're really salty, like even 10 times saltier than seawater. And so the water can get really cold and still not freeze. So Bonnie can be as -4 or -5 degrees C and still be liquid. So that's a really interesting feature of these lakes, something a little bit unexpected. Mike, do you want to go to the next? So I mentioned this, that the lakes, they have liquid water year round. And most of the time we are working there during the summer, where we can get out there with helicopter support and so on. But one of the things we really wanted to try to address was, how can we understand better what happens during the winter? So we leave in January or February at the end of our summer and walk away, and then we come back and we don't really know what happened. But interestingly, we were able to deploy some instrumentation that samples through the winter months. So for us, the winter, May, June, July. You're a little confused looking at the time scale here, but we were very surprised to see with the FLIR probe that the chlorophyll actually goes up during the winter, and there's a shift in the organisms that are contributing to that. So things are still continuing to live and grow during the winter months, even though the light goes away. But there is liquid water, and where there's water, there's life. That's one thing we've learned in the dry valleys. Want to go to the next one? Thanks. Oh yeah, so any questions either for me or maybe Mike to answer?
We've got quite a few, Kathy. I could read them out loud if you want to reply to them, since it's hard for you to read.
I'm sorry, I'm not sure if I can, yeah, I don't think I can see the chat very well. So if you don't mind reading them, that would be great.
Totally. Okay, so a couple questions about salt. One is, do the lakes have a higher salt content since the water is trapped, like in the Great Salt Lake or in the Salton Sea? And another question about the source of salts in Lake Bonnie.
Right, so it's, yes, exactly as the person who asked the question, they are closed basin lakes, and so the salts can accumulate. Like any desert environment where you have water flowing in and evaporation is the only way the water leaves, and the salts, all the solutes can accumulate over time. So that is really how the lakes have become salty. Now in Lake Bonnie, it's there's also a possibility that it was at one point a fjord, and so some of the salt that we see there was probably derived from ocean water, but also all the glaciers have a trace amount of salt that comes in like through precipitation, and so over a long enough time period with a concentration, we can build up the salt concentrations that we see in the lakes. So hopefully that answered the question. Oh, I should also say another important source for the salts is just from the chemical weathering that happens in the streams. And Anna will talk a bit more about the streams, but the glaciers have a trace amount of salts, and then that water flows through the streams and picks up more solutes along the way. So it's just about moving that material into the lakes. They tend to be the repository for everything that happens across the dry valleys, ends up in the lakes eventually.
We'll do one more question and then pass it over to Anna. Does the equipment and instruments that you use in the field have to be made especially for that environment, or can you modify things that are also used in gentler places?
Yes, both. So a lot of the instruments that get deployed into the lakes are made by companies that provide instrumentation for ocean studies. Sorry, we have a helicopter going over right now. And we do also use instruments that are deployed in lakes and so on. So it just depends on what we're trying to do and how long the instruments will be deployed. But for sure, it usually works best if we, use instruments that are built for like marine conditions, just because of the harshness of the lakes. All right.
Terrific. Thanks,
Well,
Kathy.
thank you for the question. Yep.
All right, we'll move on to streams now. Anna, are you presenting this?
Yes.
Awesome.
So Mia and I are both down here with the what we call the stream team. So we're here for the summer season, studying these glacier meltwater streams in the dry valleys. So in the summer, the glaciers warm up enough to melt, and then the melt from the glaciers flows down these well-defined stream channels and eventually flows to these ice-covered lakes. And so they usually flow for a few weeks to a few months a year. Flow usually starts in around beginning of December, and it can go until February, depending on the year. So a lot of our job, me and Mia actually went out to our first valley yesterday and opened up one of the stream gauges. The LTER operates about 17 stream gauges in 3 different valleys. And so yesterday our stream was a little too frozen, but when the streams start flowing, we have a control that measures the amount of stream flow that goes through. And so we're able to take stream flow gauge, which is height, as well as water temperature and electrical conductivity, which shows us how much ions are present in the water. And we record that on 15-minute intervals. And so along with maintaining and operating these stream gauges throughout the summer, we also take manual measurements of stream flow and stream chemistry samples from all these streams. And then we're also conducting our own graduate experiments and research on the streams as well. So the longest record in the dry valleys is Onyx River, which flows inland from the coast to Lake Vanda. And this record was started in the 1960s from New Zealand scientists. And then it was taken over in the 90s from the LTER. And the stream flow of Onyx River is pretty variable. You can see in the 77-78 season, it was close to 0, whereas in 2001 to 2002, what we call flood year, it had a lot higher stream flow. And so the large variability across years in stream flow can show us how much energy the landscape's receiving by showing us how much melt is being produced in the form of stream flow from these glaciers.
I don't know if you saw that pop up, Anna. I'll just jump in real quick. This gauge was started in the late 1960s by the New Zealand scientist group out of Christchurch, and then we took it over, so that we've had quite a bit of collaboration on this over the decades, just to keep that record going.
Yeah, so the streams connect glaciers, which you can see in the background of that photo, to the ice-covered lakes, which Kathy just told us all about here in the foreground. And along with that, the stream beds thaw in the summer, and so they have they're pretty porous. So water is able to exchange throughout the stream bed and exchange consistently through the stream, and we call that the Hyperreact Zone. And so some implications of this water exchange through the Hyperreact Zone is silica weathering, which basically just means how it breaks down in contact with water. And so we see some of the highest rates of silica weathering in the world. So you can look at that plot, and it's on a log scale, so you can tell that it's pretty high. And that is due to the sediments being pretty fresh, so there's a lot of stuff ready to be weathered, and also the short contact time, so there's only a few weeks where it's thawed and that weathering occurs and water is breaking it down. And so there is also life in the stream. So the streams host abundant algal mats. You can see here is an example of some orange and black mats. And depending on the mat type, they're located in different parts of the stream. So the orange mats are usually located in the middle where there's flow, and the black mats are located around the edges. And then the algal mats also host diatom communities, which are single-celled organisms that have silica shells. And so we can tell the type of species based on their silica shell. And so using long-term stream flow data, as well as the long-term algal mat data, we can find patterns in the diatom communities. And so this is plot is just showing that Pansia, which is the black bars in the plot, we can see are more abundant in streams that have a higher flow frequency, whereas Ludicola are more frequent in, or are more often found in streams that are more intermittent and don't have as have a lower frequency of flow. And with that, I can take any questions.
Yeah, so Anna, a couple things about streams, and then a question about kitchens. So
Okay.
About stream flow, do you notice different stream flow rates on cloudy versus sunny days? And then also, what is the typical start date of stream flow? And when do they normally stop flowing? 1 month later, 2 months?
Yeah, so it is interesting. You can tell some cloudy days that does vary in stream flow. And so it's interesting to pair that data with the meteorological data we take. And also across years, some years have more cloudy days and receive less solar energy. And so they might have lower stream flow overall. And then for flow start dates, it definitely depends on both the year as well, you know, the weather, and also really depends on streams. And so we are going to go out and open up the stream gauges over the next few weeks, and then we kind of just wait around and see when there is flow. And flow usually can happen anywhere from, I think someone was saying the earliest they saw was like November 10th. And so anywhere from like, yeah, next week or until, you know, up through mid-December. So yeah, it definitely varies by year and varies by stream.
Okay, we have a question for a budding Antarctic scientist who's concerned about how you eat down there and how kitchens work in the dry valleys when you're out in the field.
Yeah, so do you want to talk about this? Kathy has a little more experience.
Yeah. Sorry, I'll come in. Sorry, and answer that, because I love cooking, and that is one of my big concerns too, is how we eat. So we have nice shelters out in the dry valleys. We have a few camps set up, and all of them have kitchen facilities that we can use for cooking. I do want to mention that we don't have any running water out there, or you know, like all the water has to be collected either from the ice on the lakes or from glacial ice that we can collect and melt. But we do have you know, like a regular stove, and we get food from McMurdo that comes down on a ship every year. And so we have a lot of frozen food that we can use for cooking. And then people, if they're really interested in cooking, they might bring some special ingredients to make their favorite meals, like their favorite spices for something like Indian food or Thai food. So we try to get creative with the food, because cauliflower is delicious, but it's even more delicious if you add some spices to it. So, okay, that's
Perfect.
I don't know
Thanks
if
so
they
much, Kathy.
Yeah.
Yeah, perfect. The last question here is from is about differences between different valleys in stream flow, and they do tend to start at more or less the same time, but it can be staggered a little bit by sort of depending on the weather in one valley or another. And we do tend to see a lot more flow in Wright Valley in the Onyx River. It is a larger discharge in general than we see in the smaller streams, and largely that we have in Taylor Valley, and largely that's from the source of flow. Multiple glaciers contribute to the Onyx River, and typically our smaller streams only have part of one glacier that contributes to flow. So great question. Mia, are you going to be presenting soils?
I am. I'm
Perfect.
going to attempt to do a scenery change just to keep it interesting.
Awesome.
We'll
Thank
see
you.
if it tolerates. All right, so we've been talking about streams, but those streams are all surrounded by soils. So we're in this large ice-free area, and it's dominated by these sandy soils and rocky soils that we see here. And these soils are incredibly dry for most of the year. So initially they were thought to be lifeless just because they're so barren, but actually a scoop of soil in pretty much any part of the dry valleys will have some mixture of bacteria, archaea, or microinvertebrates. And even though they look pretty similar throughout all of the valleys that we'll be visiting, they actually have a pretty large amount of spatial diversity. So we have to be careful even of just like wiping off sediment from our boots and equipment as we move from site to site. And so just being careful to not introduce new species when we're moving around. And this regional biodiversity is largely a product of those different climates that we were talking about for streams. So streams will start at different dates in different valleys. And just this spatial diversity in climates leads to diversity in soil communities as well. And they're also temporally diverse. So as the season progresses and water is introduced to these systems, the communities will change to reflect that. So there's 3 main taxa that we see in the valleys. So we're seeing 2 nematode species here. There's also tardigrades, which are also known as water bears, and then rotifers. And all of these species have some form of hibernation or suspension that they undergo in between summer seasons. So they'll replace some of their intracellular water with a more sugary substance that's essentially like an antifreeze. And this allows them to shut down over the long, harsh winters and then start back up back in the summer. And so the 3 main taxa of soil invertebrates that we see are these nematodes, and we do a lot of experiments with these and just seeing how they correspond to the changing climates in our long-term record. So on the top here we have Scottnema nematodes, and these are endemic to Antarctica, meaning that they are native to the continent. And they're mostly found in low salinity soils, which is what this top plot is showing here. And then our second species of nematode on the bottom, those are hydrophilic, so they love the water. So we can see that their community and population increases as we increase soil moisture content. And this is the result of an ongoing experiment that the LTER soils team has been doing over the past 10 years, just demonstrating this relationship between the soil creatures and the water that is in the soils. So next slide. And then this is another species of nematode that we've been studying. It has a pretty interesting relationship with summer temperatures and solar radiation. So during a cooling period from the 1980s to the 2000s, 2001, we had a decrease in summer temperatures, but an increase in solar radiation during the summer. And we can see that 2 different responses of these 2 different species in this plot. And this just helps us with this long-term record to sort of project how soil communities might change as the climate changes, just seeing how each of these species responds differently to different climate and weather conditions. So next slide. So in addition to climate, the main controls on these soil communities are water and nutrient availability. And it's not exactly a straightforward answer to how nutrient limited these communities are. So this is an experiment that another experiment that the soils team did, where they essentially had plots that they were treating with carbon, nitrogen, phosphorus, and then also water. And then they compared that to untreated plots, which are denoted with the letter U, which I think is the orange line. And what they were seeing is that there's not really a uniform response when we add these nutrients and water. The big spike for the Scottnema was when they added both carbon and nitrogen, but adding each of those independently didn't really have the effect that we might have been anticipating. So definitely just needs to be more investigation and sort of discovery into how these sort of nutrients are sort of limiting life in those ecosystems. So I'm not a soil expert, but I will be happy to take any questions that you might have. Yeah.
Cool. We do have one question that came up. Are there invasive species in the dry valleys? Mia, do you know off the top of your head?
That's a great question. I actually in my previous lab was looking more at the microbial soil microbiome. So looking at possible contaminants of bacteria, and that seems a lot more likely. There definitely have been invasives introduced into mainly the peninsula, which is sort of a warmer, more hospitable environment for a lot of non-Antarctic species. So, you know, we've definitely in past years have sort of gotten more conscious of possible introduction of these species. So making sure that all of our gear and all of our boots and everything are clean when we get down here. So we're not introducing any seeds or any species that might just be hanging on when we take the flight down here. And as the climate changes, especially in the peninsula, it becomes more hospitable to those new introductions. So it's definitely been an effort by the whole U.S. Antarctic program to make sure that we're not introducing any invasives.
Yeah, terrific. Looks like we have another one here. How deep is the soil that you could dig down into? Is it frozen the deeper you dig? And
yes. So the point of refusal, which is just where the soil starts to just be sort of concrete because it's frozen permafrost soil, that will depend on site and also vary throughout the season. But for example, yesterday, Anna and I were digging down into the soil and we probably only got like 4 to 6 inches before we hit that frozen soil. So yeah, Mike, I'm not sure if you have any more input on that, but
in the middle of the summer, so our soils teams usually don't come down until January because you've got enough thaw to really have those systems functioning, and those animals come out of anhydrobiosis. They've kind of been suspended all winter, and you could probably dig down about half a meter, you know, kind of 1.5 feet, maybe 2 feet at the most. We tend to have deeper thaw where we have some saturation, right, that has made the surface soils darker. You can warm up more, you change the albedo, and you can thaw a little bit further. Sometimes those are also places that have a lot of salts. So yeah, great question there. And then one last one here: are the variety of species found in the valley found elsewhere in the Arctic or elsewhere in the world? Do you want to take that one, Mia, or do you want me to?
yeah, I can answer that sort of broadly.
Go for
So
it.
there are definitely species of soil invertebrates that are endemic. So they're only found in Antarctica, and they're pretty special to the valleys that we're studying. And that's true for diatoms as well. We have a lot of diatom or algae taxa that are specific and only found in these dry valleys. And on the microbial side of things, so bacteria and archaea, there are a lot of species that have never been described anywhere else. So there's a lot to point to the fact that these soils are pretty exceptional down here. That being said, there are species that are found here that are found pretty much everywhere. So for bacteria as well as microinvertebrates. So they would likely also be found in the Arctic, if that answers the question.
Yeah, great job.
So, yeah.
Cool. Okay, now I think I'm up to talk about the sort of the human history, and I think at the very end, the last section will be covered by Kathy. So warm up for a bit. Yeah. All right. So I'm just going to cover a little bit of the human history in the dry valleys, and really it is largely about science. It was first about discovery, and now it's about science. And these two pictures here just depict the New Zealand program back in the 70s, making flow measurements on the Onyx River with their equipment at the time, and then our more modern equipment here on the right. This is Mitch Weaver, who was a master student working with me probably 12 or so years ago, and he's making a discharge measurement also on the Onyx River. So some things stay the same, some things change. But if we go way back to the discovery of the dry valleys, Scott's men came across McMurdo Sound from their base that they had set up on Ross Island. And in 1903, they actually went up the Farrar Glacier and then came down through Taylor Valley. And what they noted was, we have not seen a living thing, even a moss or a lichen. And, they brought a lot of their perspective from the old world with them to sort of think about these places. And later in 1911, it was Griffith Taylor of Scott's Terra Nova expedition, so a different independent trip from the first one. And they noted the ice was about 6 inches thick. This is sort of on the edge of a lake or a pond, and large laminae of dull green algae practically covered the bottom. And so this is the first note of, hey, there is life out there. These are not the valleys of the dead. And so in 1903, we had this perspective of the valleys of the dead. And then of all people, a geologist came along. That's what I love to say. A geologist came along and discovered life. So that's pretty exceptional. In 1957-58, we had the International Geophysical Year. And this was a time when there was a whole bunch of discovery and science being conducted across the continent. And that included the dry valleys. In 58-59, we had this group, small group of a couple of students and professors from University of Wellington Victoria University of Wellington from New Zealand come down and spend almost 2 months in the dry valleys by themselves. They were mostly based out of Wright Valley, but they explored as much as they could, and they brought very much a geologic perspective. But then in 1961, we had quite a bit of significant lake research on Lake Vanda in particular. And so, these folks, some of the most famous US limnologists, scientists who study lakes, were involved in this work. John Hobby, Charles Goldman, Gene Likens, these are all sort of folks that have gone on to do other really incredible things, but they came down and studied and tried to understand why we saw profiles of heat and salt the way we did in these lakes. So there was true discovery there. In 1969, we talked about earlier, the first stream gauge was installed on the Onyx River, and it was installed that the Onyx flows from the coast inland to Lake Vanda, and the Onyx River the Vanda stream gauge was set up just upstream of its entry into the lake, so the terminus of that particular river. That also happened to be very close to a station that the New Zealand Antarctic program had set up for quite some time, Vanda Station. So their main base is back very close to McMurdo on Ross Island. It's called Scott Base. This was an outpost for them out in the dry valleys. On the right, that lower right, so on the lower, next, these pictures include Kiwi researchers being funny on top with a boat trying to pull it across the ice, or row it across the ice. And then in the lower right, that's the Lake Vanda from High Perch. And then in 1972, a second stream gauge was installed on the Onyx River, and it was installed way upstream near its source. So one of the things that we can do nowadays is look at the mass balance of water passing one and then the other stream gauge, and they're about 30 kilometers apart. So this is quite a long river for us to be able to look at some of these dynamics. In 1987, some colleagues of ours, Diana Freckman, or now Diana Wall, and Ross Virginia, were able to get a hold of a sample that somebody else collected for them, and just looked for microinvertebrates, and they found some. And so in 1990, they were able to get a hold of a few more samples. Actually, I think they had their own study based on those preliminary findings, came down and collected samples at Cape Royds, at Garwood, at Taylor and Wright Valley. So these are all different sort of places along the coast here. And they found abundant invertebrates. So the numbers here show as many as almost 3,000 nematodes per kilogram of dry soil. And this was, again, sort of a revelation. These were not the kinds of things that Scotsmen would have seen unless they had a microscope with them or brought samples back to look at under a microscope. But with our modern techniques and our modern understanding of soil science, we could find some of these new things. And Diana and Ross, the two authors of this particular paper with the figure on the right, they were part of the original LTER, McMurdo LTER grant and PI group. In 1993, not only with Diana and Ross's work, but also with previous work done by John Priskew in the Dry Valleys, Diane McKnight working with the USGS in the Dry Valleys, Barry Lyons working on a variety of glacial projects in the Dry Valleys. That group came together to write the first proposal that was funded by the National Science Foundation to get this whole program up and going. Now, since then, I've sort of skipped ahead and said, well, and ta-da, here we are. But there has been 30 years of evolution of ideas, of new findings, of scientific techniques that allow us to do things that are different. But often we kind of have to go back and think about our predecessors and pay a little bit of homage to them. In this case, I'm choosing to feature these two New Zealand groups. The Vandals were the name for the scientists that would work out of Lake Vanda, the Lake Vanda hut. And then the Asgard Rangers were this group of scientists that might traverse the Asgard mountain range to go from Wright Valley by foot over to Taylor Valley to make measurements of lake levels and things like that. And so we see a black and white picture on the left, a color picture on the right, but these are all our forefathers. In this case, they are all men, as it works out. Although I would say we actually have quite a wonderful gender balance on our team right now. In fact, deployed, I think we have a majority of female scientists, which is awesome. But these forefathers sort of paved a way for not only discovery, but also, a record that we can work from now. And I don't know if they knew the value of what they were doing 50 years ago or what the value we would have 50 years later, but it has been really incredible. So with that, I'll just pause. I know we're at 8 o'clock. We've been going for about an hour. We have one short section left about modern sort of camp life and researcher life, but I'll take a couple of questions, if any, come up with respect to sort of the heroic age in the history. Looks like we have one here. What sounds do you hear in the dry valleys? Is it completely silent? Oh, that is a great question. So, one of the things that I like about going to the dry valleys is that as a professor, I've been teaching all semester, fall semester. I finally get to leave, usually right after Christmas. And it brings me a lot of joy to put an automatic reply, out of office reply on my email, in my inbox. And I crawl into my tent and I can sleep. Like, nobody can sort of bother me. I can do my research day in and day out. It's sunny. It doesn't matter what time of day I necessarily do it. But there's constantly wind. So the sound you hear is often just a sea breeze coming up from the ocean, or sometimes the much stronger winds that end up blowing down the valley. When that wind isn't blowing, it's actually spooky how quiet it can get. And it's kind of weird when you're going to bed sometimes at night. All of a sudden it's very quiet and you can sort of hear everybody else in their tent, I don't know, zipping up their zipper to get in their sleeping bag or whatever. It's a little disturbing when it gets really quiet, but it's also incredibly peaceful. So a great question there. The other thing that we hear is a lot of mechanical noise. If you're in McMurdo, there are a lot of vehicles moving around. When you're in the dry valleys, you'll hear often a helicopter coming from quite a ways away because there's nothing else to interfere with it. And so you're like, I hear something that's not natural coming at you. So yeah, great question. It doesn't look like there are any more coming in, so maybe I'll pass it off to Kathy here for the last section. Kathy, are you staying warm enough to complete that for us?
We are getting pretty cold, so I think we'll get through this and hopefully get in the van. Yeah.
Perfect. That sounds fine, Kathy. Yeah, thanks for being troopers.
Yeah. Okay, so so we I mentioned earlier about the facility. So this is an example of one of the field camps out in the dry valleys. This one is called F6, and it's basically named for the stream in the Frixel Basin where it is. So there's a small hut there. Inside there's a kitchen, there's heaters, and we have a small lab area. And then you can see there's tents. So when we're up during the day and working and having dinner and so on, we're in the hut, but otherwise, like at night, we sleep in our own individual tents. Mike, do you want to move on? Oh, my face is just so cold. Get cold, sorry. So this is McMurdo, where we are right now. We refer to it as town. And you can see in the foreground, there's some dorms, and then kind of in the middle of the town is this deep blue building that's like the central facilities where we have meals, and there's a station store. There's a bunch of other buildings, and one of the other most significant ones is kind of on the right, which is the Prairie Lab, where we do a lot of our work. Right now, I think we're kind of up well, we're partway up uphill, which would be kind of in the upper right part of the photo there.
Yeah, hopefully folks can see my cursor. I'm trying to point out where you guys are right now. So
Yeah, you might see it, yeah, like we're near the area with all the fuel tanks
yeah, right above that. Yep.
Behind us.
yeah, maybe. Wow. So yeah, so Prairie Lab, as I mentioned, it's a really nice facility. Actually, there's going to be a celebration in a couple of days. It's the 30th anniversary of the opening of Prairie Lab, and so the building was opened in November of 91, and we've been in there since 93, so almost since the beginning of the Prairie Lab being established. It's you know, it wasn't really designed for a bunch of scientists with their computers, which is what most of us do now, but it's a great lab facility, really well outfitted, and we definitely enjoy coming down here and working in the Curry Lab. I think it's interesting, Mike set this slide up for us, and it's all outdoor recreation, which we all do love. You can bike, ski, etc. And I think the lower picture on the right is the McMurdo Marathon, which is out on the sea ice. Aside from those kind of recreational activities, we also have like a lot of opportunities for crafting, and I can't not mention knitting, because that's my favorite. But yeah, people ask like, well, what do you do in your free time? We don't have a ton of free time here, but when we do, we definitely make the best use of it, either outside or inside. Getting to make friends here, which is one of the greatest parts about working in Antarctica, is the people, but also definitely keep ourselves busy. Mike, you want to move on to the next one? We definitely acknowledge the holidays here, and we all look forward to it. Usually we try to really take the day off and celebrate, whether we're in McMurdo or out in the Dry Valleys. We definitely make the effort to make a special meal and really take the time to enjoy ourselves. And yeah, like birthdays that happen out in the Dry Valleys, we definitely try to make a cake and make it special for the person whose birthday it is. Here is one of our field camps. This is Lake Frixell Camp. Actually, there's a new hut at Lake Frixell that's been up for a few years, but I do have warm nostalgic feelings for our James Ways, which is what we previously used for a hut out at Lake Frixell. So the James Way and then various labs, they're small but functional. We would have one set up for filtering and one maybe for electronics, different uses in the spaces, and we can kind of spread out and do our science in those small field labs. So the facilities are actually, you know, not fancy, but totally adequate for most of what we're trying to do. Definitely supporting the science that we're doing out in the Dry Valleys. Want to move on? Yeah, and just to comment, like I mentioned back in the F6 picture, you know, it's a beautiful spot. Having your tent out in one of the field camps, having a place to go at night, kind of relax and sleep, and with a beautiful glacier in the background when you wake up in the morning and look out, it's a great place to work and live and do science.
Well, thanks, Kathy. If you guys are probably pretty chilly, so how about if I wrap up with my slides? If you guys want to go, you know, go, please definitely be warm, but we really appreciate your time and effort. I'll wrap up answering some questions and just doing some acknowledgements, but I really want to acknowledge and thank you guys for everything you've done to be out in the cold and be here for all of us online.
Yeah, thanks for everyone for tuning in and for all the questions.
You bet. All right, take care, guys. We'll be in touch. Wonderful. All right, well, I'll just wrap up with some acknowledgements before I get to these last questions. I see a few queued up on the Q&A. You know, we have to acknowledge the National Science Foundation, both for funding the work that we're doing through grants. In this case, this is our we're in the fifth incarnation of our McMurdo LTER grants. So by the end of this, we will have been going for 30 years. And I say we, I should be careful because I wasn't there on day one. I didn't come along and participate until 1998. We've had a number of field teams over the years. We typically have 31 people deploy each year. This year it's 10, and we're really grateful for that. And then the U.S. Antarctic program is sort of almost an umbrella for a bunch of contractors that help run the program day in and day out, both back here in the U.S. and in Antarctica in a variety of places. And so I just want to acknowledge a number of people who are not identified directly here. I would also note that this year we got really serious about trying to offset some of our carbon use, and in particular the footprint that we leave from taking about 22,000 miles of airplanes round trip from our homes in the US to McMurdo Station and back. And so we have individually paid for out of pocket. We haven't been using our grant funds for this, but are out of pocket to try to offset those carbon costs. And or carbon use. And when we do that, we've been specifically targeting opportunities that actually support projects that offset carbon, as opposed to sort of greenwashing and looking for, I don't know, changes, sort of more financial means and so forth. So on the ground, but in our case, we calculated this for our 10 deployers to be almost 89 metric tons of CO2 that we've offset. And that was about $150 a person or something like that. The other thing I'll just note, mcm.lternet.edu or mcmlter.edu will get you to our webpage. It has a whole bunch of data for anybody who's looking for some. It has some of our outreach activities. Go ahead there and explore, reach out to any of us if you have any questions. We'd be happy to guide you. And with that, I'm just gonna now take a few of these questions. This is an undergraduate researcher, Ken Hill, probably 20 years ago, doing some work with some GPSing around Lake Frixell here in the foreground. All right, so we have a question. What do you do with all your garbage and waste? One thing we didn't mention in the last section there is our waste that is collected in the field camps does actually get collected into 50-gallon drums, or if it's solid waste, it's divided up into a variety of different streams. Those in large part come back to the US to get processed. I know that sounds a bit inefficient, but we just don't have a number of other depots or locations where we can drop it off in between. We also have big supply ships that are going back and forth from the west coast of the US to Antarctica annually. So there is some room and capacity to specifically take on those loads. And so that's an important aspect of removing waste. The other thing I'll note is that when we're out in the field like Ken here, we carry pee bottles with us. And I don't mean to be gross, but I do just want to impress upon you that we do not just go out and leave any of our own human waste on this landscape. We collect it and take it with us and take it back to camp. And that's an important aspect of being responsible in these systems where we're trying to study the very biogeochemistry that we might be altering when we, if we were to leave any of that behind. So great question. Next one I see, are samples removed from the Antarctic and do they remain there for complete study? Great question. Kathy is actually an awesome chemist. She will run some of our aquatic samples in McMurdo. So typically that means major anions and major cations. And then dissolved organic carbon concentrations are very low, and we also run those right away. We can't really expect those shipping things back to the US takes a couple of months, and we don't think that those samples would be stable coming all the way back. They would break down and change, and so we have to analyze those immediately. Our nutrient samples, though, for example, so nutrient concentrations in stream water or lake water, we filter those and we acidify them, and then we freeze them and send those home for analysis. So a little bit of a mix there in that perspective. Thanks so much to the women in the field. Yes, they are awesome, and I'm so glad to have them down there contributing to this awesome effort. And I guess one more, not a question but a comment. Thanks to you all for this. So thank you very much for your time, Alex. I can't seem to scroll down. Can you? I can't quite see other questions. Oh, there we go. Wait.
Oh yeah, there we go. Sure.
Sorry.
Are there any programs for high school or college students for field or remote research? areas in the and of the Arctic and or Antarctica?
So for undergraduates, we do have a lot of projects will have their own what we call REU programs, which is research experience for undergraduates. When it comes to high school students, I don't know of a broad program that sort of accepts a lot of them, but if you can find a local PI, in other words, a scientist at a local university or any university for that matter, who is working in these areas, and you can, find a way to engage with them, you might be able to carve out something like an REU, an undergraduate research experience. I've done a few of those in the past, and they've been, I don't often advertise directly for those. I typically will advertise more to our undergrads at our universities, but it all depends on sort of opportunity and who has some interest. So great question. And there are, by the way, a few REU sites. So these are projects that are dedicated to having a big cohort of REUs, which occur as well. So look for those opportunities out there. Yes, and then there's high school teachers. Thanks, Rich, for putting that in. The Polar Trek program is a way of connecting with scientists and then deploying with them either in the Arctic or the Antarctic. And as you can imagine, those would be two different times of year in general, but sort of depends on the project. So that is a fantastic opportunity for teachers. We've worked with a number of Polar Trek teachers over the years, and we work really hard to sort of screen them a little bit, like this going away for 6 weeks or 2 months is not for everyone. And there are a number of creature comforts that you give up to go sleep in a tent and so forth. And so screening in large part just means, are you up for this? It's, the people who come to us and say, well, I've never been to the seventh continent. I'm like, well, okay, but what about the science of the seventh continent? That's what we're really interested in. So, so aside from that, I think, does that cover all the, oh, no, I see a couple more. I
think that's all, Mike. I think that was all. That everything else is kudos and thank yous. Awesome.
Well, thank you, everybody. I know we went over an hour. I really appreciate your attention and your ability to participate in this by asking questions. I was really happy to see my field team. We've only been communicating via email, and this year they had to quarantine for 5 days in San Francisco before and take repeated COVID tests before they could go on a charter flight to New Zealand, where they quarantined for another 2 weeks and took regular tests. And then they all got to deploy last week. I believe they arrived on the ice. So it was great for me to get to see them as well. So thank you, everybody, for coming. We really appreciate your interest and attention.